That's exactly what the name stands for. Archae, original, primitive, plastid. Chloroplasts are a type of plastid. Okay, there's other types of plastids that are very similar to each other, and a lot of times they can change from one type to another. But this here was where, in the ancestor of the Archaeplasta, that the primary event in the symbiotic event that led to chloroplasts. However, we have secondary endosymbiosis in that a eukaryotic algae, single-celled eukaryotic algae that now has chloroplasts derived from cyanobacteria, has been engulfed by another eukaryote, and we have a eukaryote living inside of another eukaryote. And another endosymbiotic relationship, and eventually the photosynthetic endokaryote also loses its identity, leaving only its chloroplast. And that has happened, we know, at least 3 times. At least 3 times. Okay, so this here is from your book that sort of shows this. Here we have an early eukaryote cell that was heterotrophic, and it engulfed a photosynthetic cyanobacteria. That became a chloroplast. Okay? It has 2 membranes. Chloroplasts have 2 membranes. What do they correspond to in a gram-negative bacterium? The outer membrane, chloroplast membrane, is homologous to what? The lipopolysaccharide layer, the outer membrane of gram-negative bacteria. The inner membrane is homologous to the plasma membrane, cell membrane of gram-negative bacteria. Okay? So cyanobacteria only have chlorophyll A. They don't have chlorophyll B. They only have chlorophyll A, and this is called a green lineage. We have a group of organisms that we refer to as green algae. Some of them can be single cellular, and eventually from them we get land plants. Land plants have evolved. They're up here, okay? In this clay, we have land plants. We also have green algae. In fact, let's go ahead and just put them up here. There's 2 different groups here that we have green algae and another branch, red algae. Okay, all right. Here we had a division point between 2 different types of chloroplasts, what we refer to as a red chloroplast and a green chloroplast. Green chloroplasts have chlorophyll B in addition to chlorophyll A. Okay, cyanobacteria don't have chlorophyll B. This was a novelty. It was derived from chlorophyll A. Okay, in the red lineage, it doesn't have chlorophyll B. It has chlorophyll C. Something different. Okay, so this created red lineage and green lineage. All right, so the green lineage, of course, led to land plants eventually. This here led to red algae, the red lineage. But also, if we have unicellular ones of these, especially of the red type, we had a second endosymbiotic happening, this time involving a eukaryote. This is what the evidence seems to suggest, a heterotrophic eukaryote. Heterotrophic eukaryote engulfs a heterotrophic red alga. Okay, so now we have okay, this is photosynthetic. If I said, let me back up here. A heterotrophic eukaryote engulfing a photosynthetic eukaryote, okay, living inside of it, and eventually the endosymbiont has lost its identity as a cell, and it is now just chloroplast. Now, this happened in the chrome alveolata, right here. That event right there. Secondary secondary endosymbiotic origin of the chloroplast. What evidence we have for this is that several. For one thing, these things, instead of having two membranes around the chloroplast, they have 3 or 4. Now, where would they have 3 or 4 membranes? Why? Okay, our chloroplasts originally had 2 membranes, but this was inside a cell that had a plasma membrane, right? Okay, so we have the original plasma membrane of the cell, and so some of these may have 3 membranes. If they have 4, it's because it has outside of it yet an endosomal membrane. You remember what an endosome is? Phagocytosis, endocytosis, plasma membrane folds in, and you have something on the inside. Okay, so there we would have another membrane. And all of the other cellular contents have disappeared. Except for some of them, believe it or not, have a little tiny, tiny functionalist nucleus that still has a little bit of DNA in it. Okay, still has a little bit of DNA. Now, this isn't in all of them, but some of them have this little defunct nucleus with DNA in it. Okay, another evidence of endosymbiosis. Okay, that makes sense. Okay, so now we also have one case over here, eugenoids. Is that this group over here? They have chloroplasts of the green lineage type, and so that indicates yet another endosymbiotic, secondary endosymbiosis of a green alga in their past. Okay, and there's some other cases here that I'm not going to talk about. These are the 2 major secondary endosymbiotic events that we know of. Okay, so the primary one only happened in Archaeplastida, and then from the red alga form, engulfment of a secondary endosymbiotic event, and from a green alga over here, another secondary endosymbiotic event. And we think that there's probably others as well, all right? So it's more complicated than what we think. Okay. And this is just, once again, another figure. I believe, I don't know where I got this from. It's probably from your textbook. This was the primary event. Incidentally, this is the green lineage over here. Archaeplastida, green algae, which will become evolve from them plants. But we have something here called glaucophytes, which are really interesting because in their chloroplasts, this is the green lineage, okay, between the inner and outer membrane of the chloroplast, they have a thin layer of peptidoglycan. You don't find peptidoglycan otherwise in eukaryotes. That's a bacterial thing, okay? So that's another bit of evidence that these chloroplasts originated from cyanobacteria, gram-negative bacteria. Okay? All right. Okay, now once we have unicellular, because the first eukaryotes were unicellular, and therefore the way in which locomotion of the cells happened to go along, most of these had either flagella or cilia at some stage of their life cycle. Few of them have lost it altogether, and all these flagella and cilia always have this microtubular structure of 9 peripheral microtubule doublets and 2 independent microtubules in the middle. This is cross-sectional structure of all flagella and cilia that happen to be found in eukaryotes. Okay, now the supergroups. These are called supergroups. 4 that we're just putting them together as a polytomy. So when I say polytomy, it is a node in the phylogeny that is not dichotomously branching. Dichotomously branching means that from that node we've got 2 branches. But here, if we have 3 or more going to the same node, it is an unresolved relationship. It's an unresolved relationship. It might be real polytomy, but more likely we don't have enough evidence to resolve it. And therefore, it is put together. Now, this happened so long ago. All of these critters have changed so much that it's hard to find too many things that they have enough in common. But nonetheless, mostly this phylogeny is based on DNA sequences, because we do have well-preserved genes that every one of them have. Okay, like all of them will have genes for DNA polymerase. All of them have genes for certain ribosomal proteins. Okay, and so we have these things that we can compare the sequences to. And so we have 4 supergroups: excavata, something called SAR, which stands for Staminophylla, alveolata, and rhizaria. Okay, and they branch. So I have that right here, okay, on this phylogeny that I put up on the board. And something called Amorphia, a branch that will have several lectures just on this group, because this is where the fungi and animals happen to be. Interestingly, fungi were once considered plants, but they really are closely related to animals, okay, more closely related to animals than they are to plants. And then Archaeoplasmata. This will be another group that we have lectures on all by themselves. So for this particular lecture, I'm only concentrating on SAR, excavata, primera, these groups. That's what we went over in lab yesterday. And then definitely for the Amorphia, we have a lot of coverage for that, because that's where the animals are. And we'll have several lectures on animals. Okay? All right, so as I said there, some of the relationships are a little bit controversial, so this is not etched in stone. But probably it's stable enough that we're going to keep it. All right, this is what your textbook does. And one of the things I do not like about these bracket types of phylogenies is it's very hard to see that this is a polytomy. This is a polytomy. These are all 3, 4 of those go to this node, right? Whereas mine is very clear. That is a polytomy, okay? All right, this is where chrome alveolata happens to be. And chrome alveolata is a large part of what used to be called protista. However, not all of it, but the bulk of it. Okay, so I'm going to go through each one of these groups, and of course, this is what I would not provide you with the names on a exam, are these major branches. The terminal ones up here, I will, okay? But not the major branches. And so you'll need to know the major branches. Okay, so excavata. Apomorphy for this group over here, I'm just going to put a green line there, but that's what I'm talking about, is that all these are unicellular. Okay, there's no multicellularity here at all. They're all single cells, but on the ventral side of the cell, they have a groove in the plasma membrane that they use to scoop up food. Okay, so it's a ventral feeding groove. Okay, and that plasma membrane, that groove is supported by microtubules on the inside. Okay, to provide it, and that's something that they all seem to have in common. 2 different clades. Paraphysalia, 1 branch right there, and main apomorphic for them, they don't have mitochondria. So they've secondarily lost it, but they have mitochondrial genes in the nucleus. So we know that they came from an ancestry with mitochondria, okay? Because mitochondria is an apomorphic for eukaryote. But the loss of mitochondrial function is anapomorphic for just Paraphysalia, right? Now, these are the guys that you looked at in termites yesterday, okay? These are the guys that are in termites. And they're all heterotrophic. They have no cell walls, and they have multiple flagella. If you remember that big jelly thing, Trichonympha that you saw yesterday, it has hundreds of flagella. That is our not silver yes.
The second line on your slide says 2 plates, and then on ours it says 3.
Okay, I can, recently 2 of them have been combined. Okay, so your 2, you change that to 2. That's right. And of course they have no cell wall. Okay, this is what made them look like animals. Okay, and that's why they were classified as animals when we just only had animals and plants. All right, but they're only found in association with animals. So you saw several of them in termites yesterday. That is a mutualistic relationship. Mutualism is a type of symbiosis. Okay, you've probably heard the word symbiosis before. Symbiosis means 2 organisms living in close association. And symbiosis can be a plus-minus relationship where one is benefited and the other one is at a disadvantage, like parasitism. That would be that type. Something that is plus-plus, where both are benefited, is called mutualism. Okay? And this is what we're talking about, mutualistic relationship here. Because the termites cannot digest cellulose. Animals don't have cellulase to be able to break it down. Fungi can, and some microorganisms can. They have the cellulase to break cellulose down. So wood has a lot of cellulose in it. And so it's the microorganisms, first of all, they get a home in the gut, nice place to live, and furthermore, they get fed, because termite gives them wood. And as they digest and reproduce, then the termite can digest them, because now they're made out of material that the